So the clickbait title, the big title out there is Lab Grown Brains. Can you explain that to me?
Yeah. So what we do is we take advantage of the potency of stem cells. So stem cells are cells that we can keep in the lab, and we learn how to induce them to specialize in different cell types of the body. And brain cells is my expertise. So we find formulas or recipes that induce these stem cells to become brain tissue. So this is a technology that is not new. I mean, scientists have been doing this for a while now. But I would say that in the past 5 to 10 years is when we really learn how to grow better tissues, meaning that they're more organized, you'll have more cell types, more diversity. And even at the physiological level, these cells start to communicate more or less as the human brain does. So it becomes a very attractive model for us to study the human brain, which is something that as a scientist, we never had too much of these opportunities. We mostly rely on animal models. So that stem cell model provides us an alternative right now.
And from what I understand, stem cells can potentially become any cell, or is that false?
That is true. They can potentially become any cells. It's just a matter of learning how they do it. And we know for some cells, but not for all. So that's why what we call regenerative medicine, it's still a very experimental field where we are learning how to do those things.
Yeah. What is it teaching us? What are you using them for?
Now, I'll give you like a very clear example from my lab. You might remember in 2015, 2016, there was an outbreak of macrocephalic babies being born in the northeast of Brazil. So nobody knew what was causing that. There is one suspicion that there is the Zika virus that might be infecting pregnant women traveling. I mean, the virus was traveling through the placenta, crossing the placenta and infecting the baby's brain. So that was the hypothesis. But how to prove that? So what my lab was able to do, we used some of these organoids and we exposed them to the Zika virus. And then we saw that the Zika virus was actually killing specific progenitor cells that give rise to the human cortex, creating a macrocephalic cortical layer, exactly as it happens in those patients. So that was what we call the proof of causation. So we showed that this was the agent causing the outbreak in Brazil, was not something in the environment, was the Zika virus. So that's one example. Most of the time, researchers use brain organoids to study the cellular and molecular alterations in neurological conditions. My lab focus on autism as well, and several types of malformations. So the connection with malformation is very easily to recapitulate in these brain organoids.